Hello, myself Rajendra Shetty, Assistant Professor at Ganpat Parsikar College of Education, Armal, Goa. Title of the unit Biomolecules and module name RNA and its types. Outline RNA and its importance, composition of RNA, properties of RNA, types of RNA that is rRNA, mRNA, and tRNA. Learning outcomes understands RNA and its importance, recalls the properties of RNA, differentiates rRNA, mRNA, and tRNA, understands the importance of RNA. Ribonucleic acid. Ribonucleic acid is a single-stranded biopolymer of ribonucleotides which plays important biological roles in coding, decoding, regulation and expression of genes. The ribonucleotides are linked together by 3' to 5' phosphodiester bonds. The strand has 5' end with a phosphate group and 3' end with a hydroxyl group. This you can see in the shown structure. Composition of RNA. RNA is a biopolymer of ribonucleotides. Each ribonucleotide consists of three important molecules, that is phosphate group, ribose sugar and nitrogenous bases. Nitrogenous bases are of two types, that is purines and pyrimidines. Purines are of two types, that is adenine and guanine, and pyrimidines are of two types, that is cytosine and uracil. Properties of RNA. RNA is single-stranded in general, but in some viruses, double-stranded RNA is present. It is more reactive than DNA and is not stable in alkaline conditions. It is easily subject to attack by enzymes. Its strands are continually made, broken down, and reused. It is more resistant to damage by UV light than DNA. Its mutation rate is relatively higher. The number of RNA may differ from cell to cell. Types of RNA. RNA in general are of three types, that is ribosomal RNA or rRNA, messenger RNA or mRNA, transfer RNA or tRNA. Ribosomal RNA. It is important constituent of ribosomes and accounts for 80% of the total RNA of the cell. It consists of single strand twisted upon itself, as you can see in the shown structure. In helical or coiled region, most of the base pairs are complementary and shows hydrogen bonding. In unfolded region, bases do not have complements. Here, purine-pyrimidine ratio is never equal. Types of rRNA in ribosomes. In ATS ribosome, the smaller subunit 40S shows 18S type of rRNA. In larger subunit, that is 60S of ATS type ribosome, the type of rRNA present are 28 to 29 years, 5.8 years, 5.0 s. In 70 years type of ribosome, the smaller subunit, that is 30 years, contains 16 years type of rRNA, and larger subunit, that is 50 years subunit, contains 23 years or 5.0 years. Year stands for sedimentation coefficient or Swedberg constant. Messenger RNA or mRNA. Jacob and Monod in 1961 proposed the name mRNA because of their potential to carry genetic information and involved in the process of translation, that is synthesis of proteins. It consists of only 3 to 5% of total cellular RNA. mRNA varies greatly in length and molecular weight. Structure of mRNA. mRNA shows a prominent coding region which codes for proteins. It is made up of approximately 1500 nucleotides. Here, in case of eukaryotes, we refer mRNA as monocistronic because this coding region codes for only one type of proteins. But the coding region of the prokaryotes, they have spaces in between the coding region, and during the process of translation, they end up in forming different types of proteins. On 5' side, there are non-coding region 1 and non-coding region 2 on the 3' side. Non-coding region is made up of 10 to 100 nucleotides and non-coding region forms 50 to 150 nucleotides. These, both the non-coding region, they do not code for any proteins. In between NC1 and coding region, there is a start codon which is AUG. And in between coding region and non-coding region 2, there is a stop codon which is normally UAA, UAG or UGA. At the 5' end in eukaryotes, there is presence of cap which is normally methylated nucleotide. Mostly it is 7-methylguanosine residues. Cap is absent in case of the prokaryotes. In place of that, there are 7 nucleotide sequences which helps in binding of ribosomes. In eukaryotes, we see polyadenylate tail at 3' end which is made up of about 20 to 200 nucleotides. This poly tail is absent in case of prokaryotes. Transfer RNA or soluble RNA. It is the smallest of 3 types of RNA having about 75 to 95 nucleotides. It forms about 10 to 20% of the total RNA of the cell. There are about 100 different types of tRNA. Each of the 20 amino acid has a specific type of tRNA. Structure of tRNA. It is a single polynucleotide chain folded upon itself to form 5' structure as per cloverleaf model. The 5' are named as acceptor stem, D-arm, anticodon arm, variable arm, and T-Sisi arm. Each arm consists of 2 parts, that is stem and loop. Stem shows base pairing between adenine in uracil and guanine and cytosine, but no base pairing is seen in the loops as you can see in the diagram. Acceptor stem of tRNA. It consists of 7 base pairs and 4 unpaired nucleotides. It shows CCA sequence at 3' end. Amino acid binds to this sequence, therefore it is known as amino acid binding site. This you can see in the shown diagram. D-arm, it shows 3 to 4 base pairs in the stem and 7 to 11 unpaired nucleotides in the loop. Loop of the D-arm is called D-loop or loop 1 or dihydrouridine loop or simply DHU loop. The amino acid activating enzyme recognition site is located on the part of the D-loop and a part of the acceptor stem on the 5' side. Anticodon arm. It shows 5 base pairs in the stem and 7 unpaired nucleotides in the loop. Anticodon loop is also called loop 2. Middle 3 bases of the loop forms anticodon which recognizes the codon of the mRNA. Variable arm. It is also called lump, mini loop or loop 3. It is of 2 types. In one type there is a loop containing 4 to 5 bases but no stem. In other type, the arm consists of 13 to 21 residues and both stem and loop can be distinguished. T-Sisi arm. It consists of stem having 5 base pairs and a loop of 7 unpaired nucleotides. The outermost of 5 base pairs is cytosine and guanine. T-Sisi loop contains a constant T-Sisi sequence. On the T-Sisi loop, there is a G-T-Sisi-R sequence which is a ribosome recognition site. Functions of RNA. rRNA gets associated with specific proteins molecule to form ribosomes. 5S rRNA and surrounding proteins complex provides binding site for tRNA necessary for protein synthesis. mRNA carries genetic information from the nucleus to the cytoplasm of the cell. It transcribes the genetic code from DNA into a form that can be read and used to make proteins. tRNA transfers amino acid to the ribosomes during the process of translation, which are joined together to form polypeptide. These are the references. Thank you.